off, we'll start by talking about biomolecules. And the first question is, what are the 4 classes of biomolecules? Which ones are polar versus non-polar? So this is tying into the macromolecules that we talked about. And what are the 4 classes here? So what were these? What were these 4 biomolecules we talked about? All right, so we said carbohydrates. What else? Nucleic acids. I think I'm hearing that over here. Nucleic acids. Lipids, right? Lipids or fats. And the final one was a pretty important one. What was the 4th biomolecule? Proteins, perfect. Proteins, okay. Now of these biomolecules, which one did we say were polar? Which ones did we say were polar? And what does polarity mean? So which of these were polar and what does polar mean? All right, polar means that it likes water, right? And which of these did we say were polar? Can anyone remember? We said one of them was non-polar, and which one was that? Lipids. So lipids were non-polar, and the other 3 were polar. Okay, and as we correctly said, polar meant that they were hydrophilic or that they liked water. Like water. Okay, what were some examples of these types of, or what was the reason rather for them being polar? We talked about that briefly. Right, they had more oxygen and this gave them partial charges across the molecule. Okay, whereas non-polar biomolecules such as lipids, and lipids include ketones, phospholipids, steroids, and things like that. And these were hydrophobic, right? Which means that they feared water. And these were charge neutral. Okay? These were charge neutral. Alrighty. Okay, let's go on to question 2. What is the goal of cellular respiration and what 4 processes are involved? What was the goal of cellular respiration? To make ATP, correct, yes. And what would we use that ATP for? What is that ATP? Energy, so it's gonna be energy for cellular work, right? So basically to harness chemical energy from our biomolecules, mainly carbohydrates, right? So from biomolecules, such as glucose, to do cellular work. And what were some examples of cellular work that we talked about? What are some things the cell had to do that it needed energy for? Transport, right? Transport of substances. Synthesis of certain substances, synthesis of hormones, enzymes, repairing DNA, all those types of things require energy, okay? And so that was the goal of cellular respiration. So we bring in our food, those are reduced to biomolecules, and then we can harness the energy from those biomolecules for the cell to do work, okay? So what were the 4 steps involved, or the 4 processes involved in cellular respiration. So glycolysis was first. That linking step, right? That linking step, where we converted pyruvate to acetyl-CoA. What else? What was the 3rd step after that linking step? The Krebs cycle or the TCA cycle. Krebs cycle. And then the final and probably the most important step was the electron transport chain, right? So the electron transport chain. Now, where did the glycolysis occur? Just to review that. Where did glycolysis occur? Not the mitochondria, the cytosol. The cytosol. The cytosol or the cytoplasm, right? What about the linking step? The linking step, where did that occur? So both the linking step and the Krebs cycle occurred in the mitochondrial matrix. And where did the electron transport chain occur? Where did the electron transport chain occur? This occurred across the inner mitochondrial membrane. Right, if we remember those proteins, those complexes that were inserted into the mitochondrium, the inner mitochondrial membrane, and so the electron transport chain took place across that membrane. Okay, that was oxidative phosphorylation. Alrighty, let's move on to question 3. Any questions on that one? Okay, what are the basic components of a biological membrane? So we spoke about some basic components that were involved in the biological membrane, the fluid mosaic model, and some of the components. What were those? Come on, this should be a review. This should come very easy at this point. What were the components of the fluid mosaic model? How about this table over here? What do you guys think? At least one component of the fluid mosaic model. You can discuss it. Just think about one. We talked about several. So we're talking about the components of the biological membrane. So we spoke about the cell membrane, right, the plasma membrane, and certain components of that, specifically certain biomolecules and how they gave certain features to the plasma membrane. So what was the base? I heard cholesterol. Cholesterol? All right. So you said cholesterol was one. Thank you. What else? Glycolipids, right? So we had some carbohydrates in the way of glycolipids. And we also had glycoproteins. What else? What was the most important component of the plasma membrane? Phospholipids, perfect. So the phospholipids, which made the phospholipid bilayer. And what was the quality or the property that we said the phospholipid bilayer gave to the plasma membrane? We spoke about a certain property that described the phospholipid. It's amphipathic, perfect. So it's amphipathic, which basically means it has areas such as the heads of the phospholipid that like water, and then the fatty acid tails that do not like water, okay? What were some other components here? Very important components. I think I'm hearing it. Proteins, perfect. So we had proteins, and we spoke about two types of proteins, two general categories. Those were peripheral. And peripheral proteins were on one side of the plasma membrane, correct? Usually the cytosolic side. Whereas we also had integral proteins, which could either be transmembrane, meaning that they go through the entire length of the membrane, or one-sided, meaning that they went through one layer of those phospholipids. Okay, so that's your peripheral proteins and your integral proteins. All right, and just to sum up here, we said that cholesterol, what property did cholesterol give to the plasma membrane? Right, good. So it added flexibility or fluidity. Flexibility or fluidity. And it prevented crystallization, okay? Right, and we spoke about some examples of proteins that were integral proteins when we looked at carrier proteins and so on and so forth, okay? All righty, let's move on to question 4. Move on to question 4. Oops. And this is where we started talking about cell membrane transport. And what are the driving forces? What driving forces act on a cell membrane, and what influences each of these driving forces? So what are the driving forces? So we had the chemical driving force, yep. And then the electrical driving force. And what influenced the chemical driving force? Or what was response? The concentration gradient. So the concentration gradient of the solute or particle in question. Okay. What about the electrical driving force? What influenced the charge, right? So the charge on the particle, something else. The charge on the particle in question, as well as the polarity across the cell. So the polarity across the cell, okay? Which is essentially the membrane potential. And then we also had the combination of both of these, which is the electrochemical driving force, which is basically the sum of the two forces. Okay. Now let's talk about determining the direction of flow when we have both these forces acting on a cell membrane or on a cell. Okay, and then we spoke about the membrane potential versus the equilibrium potential, correct? And we said that there were 3 scenarios if the chemical driving force and the electrical driving force acted in opposite directions. And what were those 3 scenarios? We said 1 was if the membrane potential was greater than the equilibrium potential. The second one was if the equilibrium potential was greater than the membrane potential. And then the third one was if they were equal. Okay. So if the membrane potential was greater than the equilibrium potential, which of the driving forces was more dominant? If the membrane potential, not the chemical, the electrical, right? So the electrical driving force was more dominant than the chemical driving force, and the electrochemical driving force follows the electrical driving force. Okay? Whereas the opposite was true if the equilibrium potential was greater than the membrane potential. In this scenario, the chemical driving force would exceed the electrical driving force, and the sum of both forces follows the chemical driving force. Okay, this is getting a bit crammed. Alrighty, and the last scenario is if they were equal, and what did we say happened if they were equal? No net movement, so I'll put no movement, and the ECDF was equal to 0. Okay. So there was no net movement, and the electrochemical driving force was equal to 0. So those canceled out. Alrighty. Any questions on that bit? Okay, let's move on to our last question, and this is what we covered more recently. What are the two main types of cellular transport, and then what are some of the characteristics of the subtypes? All right, so the first two main types are active and passive. So we had active transport versus passive transport. And what were some distinguishing properties of active versus passive? Active requires energy, or we can say uphill movement, whereas passive was spontaneous, right? Or we'll say downhill. Okay, now what were the two subtypes of active and the two subtypes of passive? So the two subtypes of active, primary versus secondary. Primary, secondary, okay. And what were the distinguishing characteristics of primary active transport? What were some unique features there? It required the use of ATP. So this is going to require hydrolysis of ATP to fuel movement. Okay, because we're moving against, our concentration gradient, we're going to require energy, and primary active transport uses ATP as the source of energy. What about secondary active transport? What was the source of energy for secondary active transport?
It uses energy from like stealing energy or whatever. Right?
Okay.
It takes energy from ions that are like coming across the big concentration gradient.
Right, perfect. So I like the way you said that. So it's like stealing energy or borrowing energy from the electrochemical driving force of another solute. So the electrochemical driving force of another solute fuels the movement of this particular ion. Okay. So in brackets here, I'll put stealing energy. It's taking the energy that's produced when another molecule is moving along its gradient. Okay. Let's talk about passive transport. So we said it's spontaneous, or it's downhill, meaning that it's not going to require energy. Right, as we saw here, it's actually going to generate energy. So what were the two subtypes of passive transport? We spoke about two subtypes here.
Diffusion.
Not diffusion. Well, simple diffusion is one. Yes, you're right. Let's make a third category here. So there was simple diffusion. What were the other two? Facilitated diffusion. And then finally, there was through channels, right? So channel movement through channels, okay? Simple diffusion was straightforward. What did that rely on? Purely on the concentration gradient. So it relied on the concentration gradient only. Okay. What about facilitated diffusion? It was also movement according to the concentration gradient, but what was the exception here?
Co-transport?
No, co-transport. What did we have to have for facilitated diffusion? Transporters or carrier proteins. So there was the use of transporters. Transporters or carrier proteins. Carrier proteins. And an example of that was the GLUT4 transporter. All right. And just to review, what was exactly happening with the use of a transporter or a carrier protein? What was happening there? What is the GLUT4 doing? It spits it out. Good. So it gobbles up that molecule or that substance on one end at the binding site. It shape shifts or has a conformational change, and then it spits that molecule out on the inside of the cell. Okay. And then finally, movement through channels was pretty much self-explanatory, right? The use of channels. And he spoke about some examples of those. They were the aquaporins, as well as we had some ion channels. And there are many different types of channels, leak channels, gated channels, and things like that, right? But the important thing here is that we have a channel or a pore or a passageway that is embedded in the membrane where certain molecules or water can move from side to side, okay? And again, these three are dependent on the concentration gradient. These are going with the concentration gradient, whereas these two are going against the concentration gradient.